Today, we are on the hunt. We're diving into a full-on scientific detective story, a quest to unravel a mystery that's been hiding inside the cells of pretty much every complex living thing on this planet.
Just think about this for a second. What if there was a protein that was so common, so ancient, that you could find it in the fungus growing on a forest floor and in the cells of your own body, but for decades, nobody knew what it actually did? Well, this isn't science fiction. It's the real story of a gene that scientists call a known unknown.
So, what exactly is a known unknown? Well, it's pretty much what it sounds like. It's a piece of biology we know is there. We see it everywhere, but we have no idea what its job is. It's this tantalizing clue that nature has decided something is so important, it's been preserved across millions of years of evolution, and it's our job to figure out why.
And our investigation today, it centers on one of these exact mysteries, a protein that has had researchers scratching their heads for a long, long time.
So let's meet our protagonist, BM46. It's a member of this huge family of proteins called alpha/beta hydrolases, which are kind of like the tireless utility workers of the cell. But here's the thing. While its family is famous, BM46 has been a complete enigma. The fact that you can find it in fungi and plants and insects and, yes, in us, tells you it must be doing something absolutely fundamental. But what is it? To crack this case, the first thing researchers had to do was what any good detective would. They had to build a family tree. By comparing the BM46 gene across hundreds of different species, they could start to trace its history and look for clues. Now, I know this looks really complicated, but what you're seeing here is basically a family tree for this one single gene, stretching across almost all of life. Every single branch is a different evolutionary path. And what's just incredible is how far it reaches. You can see these distinct branches for whole kingdoms of life. Down here in blue, you've got the fungi, over here in green, the plants, and way up top in red are the vertebrates. That's us. This picture tells us that BM46 isn't some random protein. It's an ancient evolutionary heirloom. So what did this family tree actually tell them? Well, it boiled down to 4 huge clues. First, this thing is ancient. Second, most simpler organisms get by with just one copy. Third, for some reason, vertebrates like us decided we needed several specialized versions. And fourth, and this is a big one, it's only found in complex life, what we call eukaryotes. You will not find it in bacteria. And that suggests it's involved in a process that's unique to cells like ours. Okay, so the family tree gave us the big picture, but the next set of clues came from zooming way, way in, searching the genetic code itself for unique markers that act kind of like fingerprints, telling one version of BM46 from another. And these genetic fingerprints have a name. They're called indels. It's just short for insertions or deletions. The best way to think about them is like regional accents in our DNA. You know how you can sometimes tell where someone's from by a tiny quirk in how they say a word? Well, indels do the same thing for a gene's evolutionary history. And this right here shows you exactly what I mean. What researchers did was line up the BEM46 protein sequence from a bunch of different fungi. Now, look at that big white gap in the top section. That's a deletion. And see the corresponding block of letters in the bottom section? That's an insertion. These consistent gaps and extra bits of code are the indels, and they act as crystal clear genetic fingerprints for different groups. And when they looked closely, they found these 2 really obvious patterns. The ascomycete fungi, that's stuff like bread mold, are missing this big chunk of sequence that the basidiomycete fungi, like mushrooms, have. It's a clear dividing line, a genetic signature that perfectly separates these 2 ancient fungal families. So these indels, they're incredibly powerful tools for tracing evolution. Okay, so we've traced its family history, we've found its fingerprints, but the biggest question of all is still hanging out there. What does BEM46 actually do? This, my friends, is where the real breakthrough happens. The very first major clue about its function came from figuring out where it lives inside the cell. And you can think about the answer with a really simple analogy. This protein has its own return to sender address stamped right on it. And that address is this tiny 4 amino acid tag at the very end of the protein, PEKK. You can see it highlighted right here. The researchers had a hunch that this little tag was a signal, a message to the cell's internal postal service, telling it exactly where this protein belongs, in the endoplasmic reticulum, or the ER, which is basically the cell's protein and lipid factory. And this slide is the smoking gun. It's the proof. In the images on the left, with that PEKK tag still attached, you can see the BEM46 protein, glowing green, wrapped neatly around the nucleus, exactly where the ER is. But now look at the image on the right. When they snipped off that tiny 4-letter address, poof, the protein is just lost, scattered all over the cell. That PEKK tag is an ER retention signal, loud and clear.
So now we know where it works. The next question is how it works. BM46 is an enzyme, which means it has to have a functional core, an active site that does all the work. It's like finding the essential tools on its own molecular Swiss Army knife. And here it is, the beautiful 3D structure of BEM46. All these ribbons and coils you see, they aren't random at all. This is how the protein folds itself into a very specific shape, like a piece of molecular origami. And this intricate folding creates a very precise little pocket. That's the active site, and it's where all the chemical action happens. And right inside that active site, the researchers found the 3 critical tools: the amino acids serine, aspartic acid, and histidine, all at very specific positions. This catalytic triad, as it's called, is the classic toolkit for this whole family of enzymes. So the case is coming together. BEM46 is a fully equipped enzyme located in the ER, ready to get to work. So we've cracked the case of the fungal BEM46. It's an enzyme that works in the ER. Cool. But why does this fundamental research actually matter to us? I mean, how does understanding a protein in mold help anyone? Well, it matters because that fungal version is the original blueprint. Remember how vertebrates like us have extra copies? In humans, they're called ABHD proteins. By understanding the simple ancestral version in fungi, we get massive insights into how our own, more complicated versions work. And one of those human versions is a protein called ABHD12. And this is where it all hits home. It turns out that when the human version, ABHD12, is broken because of a genetic mutation, it causes a devastating neurodegenerative disease called FARC. All of a sudden, our known unknown from a fungus is directly and critically linked to human health. Understanding the basic mechanics of BEM46 could one day help us figure out how to treat this disease. Which really just leaves us with one final fascinating thought. BEM46 was hiding in plain sight for decades. So you have to wonder, how many other known unknowns are still out there? How many other universal pieces of life's machinery are just waiting for us to decode them and unlock secrets that could be the key to our own biology and our health?